I appreciate you attending this series of lectures. I first have a few announcements. First, please, if you hear my voice, please raise your hand or send a message to me here. And also indicate in your message when you send it, where are you located now, and is this time is perfect for you, so I can have an idea when I schedule my series of lectures. Okay. I see here quite a few students. The second announcement, on Tuesday, I gave 2-hour presentation on pharmacology. We completed pharmacokinetics and pharmacodynamics. And it went very well, actually, over the 2 hours. I got the feedback from the students who attended those 2 hours, and it was very well received by the students. But I was a little bit unfortunate, but the computer freezed when I tried to save the video and process it to you. So I intend to repeat Tuesday's lecture again, and you will have an announcement for it. The purpose of this repetition is mainly to avail the opportunity for you to listen to this information, and second, I do like to be able to save it as well. I'm working on the production of the previous lectures, so please be patient with me. I actually was trying to upload a couple of files after I work it through, but it says I have to convert it to another version. So I'm working on that now, and I will pass it on to you. With that said, please give me an indication again that you can hear my voice crystal clear. Raise your hand if you can hear my voice crystal clear. I start to receive some messages here. Okay, let's move on and address carbohydrate metabolism, and perhaps give you an orientation about how we're going to proceed with biochemistry sequence. As I mentioned to you when we covered the introduction on pharmacology, I like you to understand the big picture. Pharmacology is molecular biochemistry, essentially. So we're going to address here biochemistry, and I selected this topic, but I intend, of course, to cover all of it, as well as pharmacology as well. Cardiovascular system, kidney system, endocrine system, autonomic nervous system. They all are in my great thoughts. With that said, biochemistry is very much the same as pharmacology. You're looking at the big picture here. You're looking at carbohydrates, lipids, proteins, and you're trying to understand the state of homeostasis. If you recall the statement that I mentioned it to you in the introduction of pharmacology, Dr. Minarsik in pathology addressed very superficially the term homeostasis. So here we like to address it at the molecular level. In pharmacology, we like to address it at the molecular level. In endocrinology, we'll do very much the same. And with physiology, all physiology will do with this concept. It's very important to appreciate the fact that in order for life to continue on, you must have a stable homeostasis state. So I visualize macromolecules in a form of carbohydrates, nucleotides and nucleic acids, proteins and amino acids, lipids, as several contents here. And then you see here enzymes are there, and enzymes are involved in all of those metabolic pathways. Vitamins, minerals play an essential role in all of those metabolic pathways. So that brings us back to the chess game approach. We want to understand this interrelationship among metabolic pathways that take place in different tissues. We want to understand metabolic pathways that take place in different parts of the cell. And we want to understand their regulation. We want to understand how we achieve the state of homeostasis when we address all of those decent metabolic pathways. The best way I explain it to you here, if you look at the cell, there are certain metabolic pathways takes place in the cytoplasm, others takes place in the mitochondria, and the third class that requires the collaboration of the cytoplasm and the mitochondria. For you as a professional student, you have to appreciate the locations. You have to understand and appreciate metabolic pathways, not memorize it. When I teach biochemistry to medical students and pharmacy students, majority of them actually had biochemistry before at undergraduate level. And at University of Michigan, where I used to teach, we used to accept the students with a B or better who took biochemistry under University of Michigan system, because we know exactly how to orient the students. But then they tell me, well, it's completely different. Why? Because we insert in detailed clinical applications. We insert in detailed information about homeostasis process, regulation process, interrelationship among metabolic pathways that take place in different parts of the cell and takes place among different tissues. So if you look at the cytoplasm, glycolysis takes place in the cytoplasm, lipid biosynthesis takes place in the cytoplasm, cholesterol biosynthesis takes place in the cytoplasm, nucleotide metabolism takes place in the cytoplasm. In the mitochondria, the main cycles are citric acid cycle, or TCA, as you know it, electron transport and oxidative phosphorylation, fatty acid oxidation takes place in the mitochondria. But there is an interrelationship between the cytoplasm and the mitochondria. So we have to have a full understanding of that. We have to have a full understanding of the regulation and the clinical problems that can take place by a deficiency or a mutation of an enzyme. And that's what we like to concentrate on. Urea cycle and gluconeogenesis, sorry about that, I missed to include gluconeogenesis here, requires the collaboration of the cytoplasm and the mitochondria. There must be a reason for that. See? There must be a reason for that. Urea cycle and gluconeogenesis requires the collaboration of the cytoplasm and the mitochondria, which means certain enzymes are located in the cytoplasm, others are located in the mitochondria. When you have an appreciation about these principles, it's very easy for you to understand clinical problems. What is happening if an enzyme located in the mitochondria is mutated or is deficient, and why? So that's in terms of location of the cycles within different parts of the cell. Let's address the second picture here and try to appreciate interrelationship among metabolic pathways that take place in different tissues. They talk to each other. They understand the language. Everything is there for a reason. In the liver is a factory of numerous activities. In the liver, you carry on glycogenolysis, you carry on gluconeogenesis, in the adipose tissue, you carry on essentially lipolysis that gives you an end product of fatty acids, and that can feed into the liver. You're producing ketone bodies by complete oxidation of fatty acids. Those ketone bodies can be transported to extrahepatic tissues. The muscle tissue here is able to carry on glycogenolysis as well. So there are certain enzymes in the muscle. The name of the enzyme is very much the same as the one which exists in the liver. But a deficiency of an enzyme that involves in glycogenolysis in muscle tissue may not be a life-threatening case. You can be fatigued, you can be weak, but you're not going to lose your life. Versus there are some key enzyme deficiency in glycogenolysis. If that takes place in the liver, it's a life-threatening case. So we like to enjoy this material together. We like to make sense out of it. So in the first two lectures, we like to devote it to many metabolic pathways in carbohydrate metabolism. Emphasis will be on clinical applications, regulations, but I urge you to understand specific steps. In order to do that, you have to go through the structures. At USMLE exam, you will not see structures of intermediates. But unfortunately, in order to solve a problem, you have to have 100% understanding of the specific steps. So go over it one time, and that will help you a lot to look at the big picture. When we talk about carbohydrate metabolism and glucose metabolism, you essentially have to appreciate the fact that glucose metabolism is defective in very common metabolic diseases. Obesity, for instance, diabetes, major problems with carbohydrate metabolism. Obesity and diabetes contribute to development of serious medical problems that include atherosclerosis, as you know, hypertension, vessel diseases, kidney diseases, unfortunately, blindness. We will have a complete lecture on diabetes, type 1 and type 2, glycosylated hemoglobin, after we complete this sequence. What we like to achieve when we cover carbohydrate metabolism, the objectives that we like to achieve is to be able, in a simple term, to define what's meant by glycolysis, gluconeogenesis, glycogenolysis. Glycogenesis, have an appreciation on it, and I give you some hints on that. Give an overview about glycolysis, understand it more than memorize it. Of course, appreciate and understand the physiological significance of aerobic glycolysis versus anaerobic glycolysis. Aerobic glycolysis that take place in tissues that has mitochondria. The brain has it, heart has it, liver has it. Anaerobic glycolysis takes place under anaerobic conditions. Retina of the eye, red blood cells, kidney medulla carry on anaerobic glycolysis. Must be a reason for that. Is anaerobic glycolysis is essential for aerobic glycolysis to continue on? Those are the type of questions that we like to address. Communication between red blood cells, liver, between muscle and liver. You have to appreciate the fact that the brain alone consumes 120 grams on a daily basis, and the preferred substrate is glucose, is not ketone bodies. You're causing more harm than good, actually, if you exceed the level of ketone bodies in the brain. Ketoacidosis. And that's one of the criteria that you get ketone bodies by following Atkins' diet, actually. When we look at the glycolysis, we like to appreciate the two stages of glycolysis, the prep phase, preparation phase. The main significance of glycolysis is production of energy, essentially. You're breaking down glucose under aerobic or anaerobic conditions to produce energy in the form of ATP. And that's what we like to understand and address. So you must have a prep phase and payoff phase, just exactly like what you're doing now. In medical schools, that's a prep phase. You pay off you pay your tuition, you spend the time, you struggle with the material, you pass it. That's a prep phase. The payoff phase, when you start to work as a practitioner and get your reward. That's a payoff phase. Have an appreciation about all the reactions of glycolysis. Why is that? There are some key reactions in glycolytic pathway. Those key reactions are under strict control. They are under strict hormonal control and allosteric modulation by small molecules that can include in the product, include other metabolites from other metabolic pathways. So in order for you to understand this big picture, you have to define clearly the key steps, the regulatory steps, and then rationalize how those key steps are regulated by insulin, epinephrine, and glucagon, how those key steps are regulated by metabolites from other metabolic pathways. If you understand this concept, you should be able to understand the clinical problems associated with a deficiency or a mutation of certain enzyme. And we're going to address that with a great interest to you, so you can love this material. Now, each enzyme that catalyzes each reaction. Why is that? Because you essentially, when you cover glycolysis, you also covering gluconeogenesis. And in order to compare and contrast between the two cycles, you have to appreciate the names of the enzymes, especially those ones that control the key reactions. Understand and appreciate the physiological significance of anaerobic glycolysis. We're going to understand that anaerobic glycolysis is needed for aerobic glycolysis to take place. Anaerobic glycolysis is needed for those tissues that lack the mitochondria. They have enough sufficient energy to carry on their activities. But the existence of the mitochondria may cause more harm than good. Cornea of the eye is one of it. If you have a mitochondria, and you tend to scatter the lights. Red blood cells, you have sufficient amount of energy in order to maintain the activities of red blood cells, including glutathione in a reduced form and oxidized form in the right ratio. When we address catabolism of glucose and glucose catabolism under anaerobic conditions in humans versus in yeast, we're addressing it in yeast in order to understand catabolism of alcohol. Not because I want to add a burden on your shoulders. That's not the objective. But to understand beneficial effects of alcohol at moderate consumption versus what's going to happen when you consume alcohols at high level, and why at the molecular level. Of course, know how the key steps of glycolysis are under strict regulation. Why? Because you want to see the state of homeostasis, understand how hormones control the glycolytic pathway. Why? Because you want your main objective is achieving homeostasis state. You do not want to disturb this beautiful system. Do you like this picture first? You love it? Clear. That's good. Good. Thank you. Thank you. Thank you. So I got here all the responses. Says the presentation is clear, great. We appreciate it. And I appreciate having you in this lecture here. Let us, if you cover this slide, if you understand this slide, you got it made. That is carbohydrate metabolism. So, going over the definitions. As the name implies here, glycogenolysis, breaking down of glycogen. Glycogenesis, building up of glycogen. So every time you see enesis, lysis, cleavage. Enesis, build up. Glycogenolysis and glycogenesis mechanism regulation is considered a model for hormonal control. If you understand that fully, you shouldn't have any problems with lipids. You shouldn't have any problem of putting the pieces together. Both cycles, glycogenolysis and glycogenesis, take place in the cytoplasm of the cell. Glycolysis, breaking down of glucose in a 10-step process to produce pyruvate as an end product. Glycolysis takes place in the cytoplasm. Aerobic or anaerobic glycolysis takes place in the cytoplasm. What you have to appreciate here, the entrance of glucose into different tissues is regulated by a specific GLUT transporter. So in red blood cells, for instance, you have GLUT1. And then in the liver, in the muscle, you have GLUT4. In the brain, you have GLUT3. So we will see the requirement of those GLUT and how they vary. You will see that certain enzymes are located in the muscle versus located in the liver, but one have a low affinity toward the substrate glucose versus the other one has a high affinity, hexokinase and glucokinase. And what's that mean in terms of state of homeostasis? The end product of glucose breakdown in the cytoplasm is pyruvate, and then pyruvate under anaerobic conditions in humans is reduced to lactate. Of course, there is certain enzyme here. We're going to address it, lactic dehydrogenase enzyme. Same applies in yeast, but we're going to cover yeast as a way to understand catabolism of alcohol and as a way to address the question, is drinking a little bit of alcohol beneficial? What are the harmful effects for drinking high level of alcohol, and why? Complete oxidation of pyruvate into carbon dioxide and water requires the collaboration of serious enzymes. Those enzymes are located in the mitochondria. Why? Why is that? Why you carry on complete oxidation of pyruvate into the mitochondria? Because your main objective is to produce energy. The mitochondria is the site, is the place where you have this factory that can produce energy in a form of ATP, and that is electron transport system and oxidative phosphorylation. The stage here where pyruvate undergo oxidative decarboxylation to produce acetylcoenzyme E is considered the linking bridge, just like London Bridge in London, and is catalyzed by pyruvate dehydrogenase, as we're going to see very soon. This is the complete map for oxidation of glucose produced from glycogenolysis all the way to carbon dioxide and water. You're going to understand if you carry on this cycle, all of it, glucose all the way to carbon dioxide and water, you can produce actually 32 ATP. If you carry it on under anaerobic conditions, you can produce only 2 ATP. So majority of the rest of energy is in the mitochondria. When you address glycolysis, you have to keep in mind gluconeogenesis. You see this one here, gluconeogenesis. So gluconeogenesis is building up of glucose from pyruvate. When you address glycolytic pathway, you almost address 60%, 60 to 70% of gluconeogenic pathway. Why is that? Because it has common enzymes. You can shift it either way. Depends on the substrate. Is your main objective is to produce energy, or to utilize a non-carbohydrate metabolite to produce glucose? Okay. And those cases happen under severe starvation. You're probably aware of the starvation that takes place in Ethiopia, and when you look at those pictures of the young boys, the young men, you see them extremely fatigued. And that's the situation if you starve for a severe, long period of time. You tend to utilize your proteins as a source of energy, and that is the last resort after you exhaust lipids, after you exhaust carbohydrates. We will see that gluconeogenesis requires the collaboration of the mitochondria and the cytoplasm. And it's not like glycolysis that take place, all of it, in the cytoplasm. So again, I'd like you to appreciate this chess game approach here. Last but not least, there are some side metabolic pathways that take place, like phosphopentoase pathway. You can convert glucose into 5-carbon atom sugar, and that is ribose. We will address the physiological significance of this cycle in terms of production of reducing agents to build up fatty acids, in terms of production of sugar moiety to build up nucleotides and DNA structure, RNA structure. The interesting aspect that I like to address it to you here before we move on to the details of carbohydrate metabolism. When you look at the diversion of glucose to another cycle, or the diversion of pyruvate to other cycles, anaerobic conditions, you have certain enzymes here, and you have to have a tight regulation on those enzymes. That's what we like to understand. Equally significant, we like to understand the abnormalities that can take place by a deficiency of one or more of those enzymes, and understand the interrelationship that take place in different tissues. You're going to enjoy that. For instance, a question can be asked: would you expect to see a patient who have a mutation on the key enzyme that divert glucose from its main metabolic pathway, and that is glycolysis, to phosphopentoase pathway? If you lose control of this enzyme, what are the clinical application for this? What can you get out of it? So if you tend to produce a mutant enzyme, you understand this. When Dr. Minarsik covered it, I attended his lecture on HIV and immunology. Enzymes means a lot. So we're going to understand that if you have a mutation on with PRPP synthase, one of the key enzyme, or glucose 6-phosphodehydrogenase here, you may expect to see the patient suffering actually from hyperuricemia, and why. So we're going to elaborate on that later on, but this is only an appreciation about how you look at this slide. Normally, when I cover this to medical and pharmacy students, I tell them, well, if you understand this very well, you shouldn't have any problem actually addressing any question in step 1 related to carbohydrate metabolism, or in any addressing any clinical cases. What's left for us is to dissect those at the molecular level. So I visualize that with this decent slide here that I intend to share it with you. I visualize carbohydrate metabolism as Hawaiian Islands. and carbohydrate metabolic pathways are those islands here. So the big island represents glycolysis because it's very important, and then probably Kauai or the island of Maui represent glycogen metabolism, Oahu represent galactose metabolism, and Kauai represent phosphopentose pathway. So let's concentrate a little bit on the big island. I urge you actually to visit those islands. I lived there for 4 years and was very enjoyable for 4 years. With that said, we like to look at glycolysis. What are we dealing with glycolytic pathway here? You're dealing with breaking down of glucose all the way to pyruvate. So the way you're going to see that, there are mainly 10 steps here. And you see those arrows, some of them are in red, others are in white. What do we mean by that? Essentially, what I'm telling you here in a simple terms, those are ones in red are the key steps. Those are ones in red that they must be tightly regulated. So as we're going to see, hexokinase, PFK, fructose 1,6 phosphokinase, pyruvate kinase, those are key enzymes in this cycle here. The remaining ones here in white, they're not key enzymes. They can carry the reaction in both ways. Those key enzymes carry the reactions only in one direction. So once glucose gets into the cytoplasm, it's committed to continue on. It can go on to glycolytic pathway, or it can be diverted to phosphopentose pathway. So this first step here must be under tight control. Then we're going to learn phosphofructokinase enzyme, which is the most crucial enzyme in glycolytic pathway, pyruvate kinase enzyme, and what is the problems, deficiency of this enzyme, what kind of diseases you expect to get, and why. The remaining ones here, they're not specific for glycolysis. The remaining ones here, you will see them again in gluconeogenesis. See that? But of course, in order to reverse this reaction here, you must have a different enzyme in gluconeogenesis. In order to reverse this reaction here, you must have a different enzyme. And in order to reverse this reaction, you must have a sequence of enzymes. Some of them are cytoplasmic enzymes, others are mitochondrial enzymes. So you're going to love that. But appreciate the principle first. Good, good, good. Let's look at this beautiful slide here. The glucose coming from extracellular fluid gets into the cytoplasm cell membrane by a specific receptors, and then gets phosphorylated. Once glucose is phosphorylated, this phosphate group here will make this molecule impermeable to cell membrane, so it cannot go back. It's committed. It has to go on. GLUT system, function of insulin. In certain tissues, insulin facilitates the process by which glucose can get into the cell by enhancing production of GLUT, which is the main transporter for the glucose. So we will see that glucose can get into the cell by a process of facilitated transport or co-transport in some cases. When you look at this cycle here, that clearly explains which cycles takes place in the cytoplasm, which cycles takes place in the mitochondria, which cycles require the collaboration of both. So again, as I intend to repeat it to you, glycogenolysis, breaking down of glucose, you're breaking down all glucose to pyruvate. Those three red arrows here is a clear indication about the key steps. All of those reactions take place in the cytoplasm. Anaerobic glycolysis to produce lactate takes place in the cytoplasm, but in different tissues, of course. The lactate, which is formed in the muscle, can be transported to the liver. Okay. We'll cover those cycles. And these can be converted under anaerobic conditions to ethanol. TCA takes place in the mitochondria. Let's look at gluconeogenesis. It's listed here in the mitochondria and in the cytoplasm. Why is that? Because in order to reverse this step, in order to come from pyruvate to phosphoenol pyruvate here and carry on glycolysis, this one here requires 4 enzymes. 2 of them are located in the mitochondria. The other 2 are located in the cytoplasm. And we'll cover that when we address gluconeogenesis. Last but not least, you can see phosphopentose pathway is coming from glucose. Understand, appreciate interrelationship that takes place in different tissues. Very important. You see here, you're producing alanine in the muscle. Alanine can be utilized as a metabolite to build up glucose. Why is that? Because alanine has 3 carbon atoms. So you can join the trioses here. On the other hand, fatty acids that are produced by lipolysis of fats can produce ketone bodies via the liver. So you go there. And ketone bodies can be used also as a source of energy and can go on to extrahepatic tissues. Okay. Beautiful presentation. That gives you all what we like to say. At the end of this course, you have to clearly define specific metabolic pathways that take place in different tissues and the significance of this interrelationship. The liver is the factory of everything. You see here, you're carrying on protein synthesis, you're carrying on glycolysis, you're preparing amino acid degradation to urea cycle, and urea cycle will require the collaboration of the mitochondria and cytoplasm. Both of them are there in the liver, no problem with that. Glycogenesis is needed, but then we like to fish out in some detail here. What is the function of insulin? how insulin can enhance this process via portal vein. What do we know about chylomicrons, how they are formed, how VLDL is formed, how LDL is formed, how IDL is formed, and very recent studies. I was attending a conference just a few weeks ago, and I will share with you some of this information about HDL and LDL. That's a very important topic. You see here, lactate goes from red blood cells to liver. Why? Because you can use it to produce pyruvate. See, it's very effective here. We'll cover actually VLDL, metabolism of chylomicrons. You see this brain here. You need at least 120 grams of glucose on a daily basis. If you study more, you need more glucose to keep your brain active. Brain has mitochondria, so you're carrying on this beautiful presentation. Fats that are produced in the liver, where is it going to go? You see, boop, goes to adipose tissue here. Okay, so you can see this. Spend some time and appreciate this beautiful slide here. In terms of specific GLUT system, so you have GLUT1 here, can accept glucose in red blood cells, easily facilitate its entrance to the cytoplasm. You carry on glucose 6-phosphate, you're producing lactate. And lactate is essentially producing here 2 electrons, as we're going to see. In the brain tissue, you have GLUT3 system. You have a mitochondria, so that's where you have that cycle here. And I can illustrate to you all the adipose tissue, liver, and stuff like that, but maybe later at the end of this topic. Let's take a short break, 10 minutes break, and come back and address at least glycolytic pathway, hormonal control, key enzymes. Please, 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 I would appreciate it highly if you send me your comments about the presentation. I have here some questions. I'd like to address it from some. Yeah, it says clear. Thank you, thank you. That's good. That's good. So I have that. This is a good time, but possibly 1 p.m. is better. Well, yeah, I can see your point. Maybe in some cases, but I prefer 2 p.m. because I do my regular exercise in the morning. Sometimes I stay up to maybe 11. So that gives me some time to make sure that is the presentation clear? Yeah, please give me your thoughts, and maybe after the lecture you can email it to me. And please pass the word. Did Dr. Minarsik presented this? He made the announcement. I wasn't able to attend his talks this morning because I got quite busy. But I like you to get the benefit of it. Okay. And later on, I will avail the opportunity for you to interact with me. And so it will be lots of fun. Okay, let's take a break and come back. Try to enjoy this beautiful presentation. So until I see you